Hi, I'm Amy Said from University Tunku Abdul Rahman, Malaysia. Today, I would like to share some insights into my project entitled Preliminary Toxicity Evaluation of Novel Quercetin 4' Cinnamate on HT29 human colon cancer cells. Quercetin belongs to a large group of antioxidants termed flavonoid. It is found ubiquitously in a broad range of fruits, vegetables, herbs, and wine. The desirable beneficial properties of quercetin are attributed to its antioxidative, antimicrobial, antiviral, anti-inflammatory, and most importantly, anti-cancer properties. Despite its numerous beneficial properties, its low solubility and bioavailability and thus efficacy may be limited due to conjugation reactions to the free hydroxyl groups surrounding the molecule, such as H3', 4', 3, 5, and 7-OH hydroxyl. Efforts have been made to improve the bioavailability, including to improve metabolic stability and intestinal absorption. This can be done or achieved by modifications of their structure by chemical pathway or enzymatic pathway or chemoenzymatic pathway. In this study, quercetin was chemically isolated with cinnamic acid to produce the novel compound quercetin 4' cinnamate. Cinnamic acid was chosen because, firstly, cinnamic acid can be found naturally in cinnamon and resin. Therefore, it is expected to have fewer side effects than other chemicals. Secondly, cinnamic acid itself has been shown to possess antioxidative and anti-cancer properties. Therefore, the isolated analog generated may have synergistic anti-cancer properties. Our previous findings on quercetin 4' cinnamate show that this compound had enhanced lipophilicity and stability as compared to the parent compound quercetin. The RF value was increased by 118%, and the stability in a human gastrointestinal tract stimulation was increased by 63%. On another note, this compound also showed consistent stability across the storage period up to 30 days. With enhanced lipophilicity and stability, it is therefore of pharmacological interest to evaluate the toxicity of this novel compound on HT29 human colon cancer cells. Cell viability assay, MTT, was applied to investigate the cytotoxic effect. Fluorescence microscopy and flow cytometry was used to investigate the apoptotic effect. The concentration range was set at 12.5, 25, 50, 100, and 200 micromolar. Treatment duration, 24, 48, and 72 hours. From MTT assay, the IC50 of quercetin was 158.3 plus minus 4.8 micromolar, whereas the IC50 of quercetin 4' cinnamate was 118.8 plus minus 22.4 micromolar. Quercetin 4' cinnamate had a significantly lower IC50 value as compared to quercetin. Therefore, it is fair to say that quercetin 4' cinnamate was more cytotoxic than quercetin on HT29 cells. Cinnamic acid and the solvent vehicle DMSO plus 10% twin 80 had no significant effect on HT29. From flow cytometry, it can be seen that both quercetin and quercetin 4' cinnamate induce HT29 cells to enter early apoptosis and late apoptosis instead of necrosis. There were no significant differences between quercetin and cinnamate at 100 micromolar. However, at 200 micromolar, there were significant differences between quercetin and cinnamate after 48 and 72 hours of treatment. At 100 micromolar, across all durations, there were no significant differences between quercetin and quercetin 4' cinnamate. At 200 micromolar, the reduction in the population of viable cells induced by quercetin was significantly higher than that of quercetin 4' cinnamate. Similarly, the increase in the late apoptotic cells induced by quercetin was also higher. Therefore, we can conclude that quercetin was causing more cell death in HD29 cells by apoptosis than quercetin 4' cinnamate. This finding was in agreement with a study by Landis in year 2008. By modifying the structure of quercetin by acylation with cinnamic acid, the resulting acylated analog could lose the ability to interact with cellular proteins, thus resulting in less apoptosis of cancer cells. In conclusion, even though the lipophilicity and solubility of quercetin were improved by the acylation reaction, there was a delicate balance in which the permeability of the compound to cross the cell membrane was restricted by the bulkiness of the resulting acylated analog, causing it to lose the ability to bind to the downstream modulators of apoptosis. Taken together, quercetin 4' cinnamate synthesized in this study exhibited enhanced cytotoxicity against HG29 human colon cancer cells. This warrants further investigation into the underlying non-apoptotic mechanisms and its therapeutic potential as a chemopreventive agent. Thank you.